A murder, a mystery. Anna Garcia lies dead on the ground, strangled to death. Who could have done this? We only have one clue that leads us to the cold-hardy killer. When fleeing the crime scene, the killer left a single strand of hair containing his DNA. Forensic scientists can extract this DNA and run a test to profile who the person is that it belongs to. However, one of the main problems is that there isn't enough to go around. You see, in order to be accepted into court, forensic scientists must run several tests in order to ensure that their findings are accurate. So, they must produce more copies of the DNA. This can be done by going through a process called Polymerase chain reaction, or PCR. This can be used to produce large quantities of DNA when only a small amount is available. PCR involves a repeat procedure of 3 steps in a thermal cycle: denaturation, annealing, and elongation. Denaturation involves the separation of the DNA strand. The DNA sample is heated to separate it, which gives a single strand template for the next step, annealing. In annealing, the reaction is cooled so the DNA primers can attach to the opposite ends of the strands. Finally, in elongation, a heat-tolerant enzyme called DNA tag polymerase copies the strands, producing 2 new copies of the original DNA sample. After doing this process 30 times, we would have a total of 2 to the 30th copies of DNA, which is plenty enough to use. So we have the DNA to work with, but how does that tell us who killed Anna Garcia? Well, first we'll need to look at the suspect's DNA profile. Everyone has a unique set. I do, you do. This makes it easier for us to tell the results of this next technique called gel electrophoresis. In gel electrophoresis, we look at different DNA fragments and we can compare the ones we found at the crime scene to any of the suspects that we have. First, in gel electrophoresis, the fragmented DNA samples are placed in wells of agarose gel. They are covered with a buffering solution and have an electrical current pass through them. The negatively charged DNA will then move towards the positive side, which is the bottom. Depending on the size of the fragments, the DNA will be more towards the top or bottom. Scientists would then compare the location of these fragments between suspects and use it to identify an individual. It is essential that when conducting this test, our suspect needs to have the same results as the DNA at the crime scene. Both PCR and gel electrophoresis can be used by scientists to help identify a culprit in a crime scene. But it doesn't have to be just the culprit. It could also be a victim, or anyone that was around the crime scene at the time. There's a lot of possibilities. The results are in from the test, and we have our culprit, Elena Garcia, the victim's sister. Until next time, everyone.